Today we are going to talk about G protein coupled receptors. G protein coupled receptors, which are also called 7-pass receptors and which are also called serpentine receptors, because they 7 times passes through the membrane and look like a snaky shape, like a snake, right? So wherever in the literature, medical literature, you study G protein coupled receptor or 7-pass receptor or serpentine receptor, they are one and the same thing, right? But before really in this lecture, what we will be doing? First of all, we will discuss about the structure of the receptor, how this receptor is structured, what are important domains like, right? And then we'll talk about activation and function of the receptor. After that, we'll go how these receptors are activated and how these are inactivated, right? And what is the difference between inactivation of receptor and desensitization, and how receptors undergo desensitization and how receptors can be resensitized. And then we will talk about down regulation of 7-pass receptors and up regulation of 7-pass receptors. Because why I included especially this component, because many students get confused about inactivation of receptors or desensitization of receptors or down regulation of receptor. But today we will try our best to make it crystal clear differences among these conditions, right? So before really I discuss G protein coupled receptor, let me discuss about basic concept that what is receptor, right? What is receptor? Receptor is actually, there are two types of receptors. They are sensory receptors which are related with especially with the neurological system, right? There are sensory receptors like taste buds, like rods and cones, right? Like Pacinian corpuscles. These are sensory receptors. We will not talk about these receptors today. We will not talk about sensory receptors. Today our discussion is about those receptors, those macromolecules, right, which are specially related with physiology, biochemistry and pharmacology, right? So we will limit ourselves to those receptors which are specially involved in biological system related with physiology, biochemistry and especially pharmacology, right? Now, what is receptor? Receptor is any macromolecule. What it is? It is any macromolecule or complex of macromolecules. Complex of macromolecules. Right? In old time, we used to say that such receptors are generally protein receptors. Yes, this is right. Generally, these receptors are proteins or peptides, but some of the receptors are not proteins or peptides, like DNA. DNA is a macromolecule. Of course, it is not protein. And some of the drugs directly act on the DNA. For example, alkylating agents, right? Anti-cancer drugs. Some of the anti-cancer drugs directly act on the DNA. So for those drugs, DNA itself is a receptor, right? That is why now we have changed the definition. We don't say that receptors are proteins. We say they are macromolecules, which may be proteins, which might be DNA, or that might be RNA or any other big molecule, or complexes of big molecules. Is that right? So receptors are macromolecules or complexes of macromolecules which have special two functions. They should have at least two functions, or rather I should say two functional domains. They should have two functional domains, right? Two functional domain mean there should be one domain. For example, if this is a receptor, let's suppose this is a receptor, right? And okay, now if this is a receptor, one site of the receptor, one domain of the receptor should be such that where a specific molecule can bind. And that molecule which can bind here, let's suppose this is the molecule which can bind here. Such molecule would bind with a special domain, specific domain of a receptor. Such molecule is called ligand binding domain. What is this? This is ligand binding domain. How do we define ligand? Ligand is a substance which binds with the receptor and brings a conformational or functional change in the structural and functional change in the receptor to produce some biological response. What is ligand? Ligand is the substance which binds with the receptor. Where it binds with the receptor? At the ligand binding domain. At the ligand binding domain, right? And once it binds here, it is just like this. You pull my ear, and might be kick you if you pull my ear. So your hand is the ligand, my ear is the ligand binding domain, and the kick I do or response I do, that is biological response. Yes, is that right? Okay, for this simple thing, we can say when ligand bind over here, right, whenever it brings its tongue and start tasting the things around. So it is other functional domain. First domain was ligand binding domain, right? Second domain is effector domain. What is it? Okay, I will make it like again. This is, okay, specifically, what is this? Ligand binding domain, right? Where you can bind some ligand. Now this ligand may be a hormone, maybe a neurotransmitter, maybe a drug, right? Yes. So hormones or neurotransmitter or drugs, they usually go and bind with the receptor at which domain? Ligand binding domain. And in response to that binding, receptor undergo a conformational change under structural and functional change. It undergoes a modification, and result of that modification will be that when it binds, ligand binds, and other domain is activated, right? Yes. Or inactivated. There is some change, right? This domain is called effector domain. Effector domain. So basically, what is receptor? Receptor is a macromolecule or a complex of macromolecule, right? Yes. Which should have at least two minimum functional domains, two functional areas. One, ligand binding domain, other, effector domain. You remove it, tongue goes in. You put it here, tongue comes out, right? So you understand it? Yes, so is that clear? Now, I will ask a question to you. Plasma proteins, they bind many things. Plasma proteins like albumin, pre-albumin and others, they bind many hormones, and even they bind many drugs. Are plasma proteins receptors? Yes, doctor. They are receptors. Okay, what do you think? They are receptor or not receptor? Say yes or no. No confusion. No, sir. No, why they are not receptors? They are binding a substance. They don't have effective. Excellent. He is saying that plasma protein, okay, let's suppose here is the plasma protein. Plasma protein may bind many things, but it does have binding domain but does not have effector domain. Plasma proteins do bind many substances, but plasma proteins, once they bind a substance, usually they don't undergo any conformational change, and they don't have any effector domain. And usually when substances bind with the plasma proteins, they do not produce any special or specific biological response. Yes, so it is just like that plasma protein is that you are doing like this, but there is no response. So everything which in our body, every macromolecule in our body which can bind something, right, but does not undergo any conformational change and does not produce any effector biological response, that thing cannot be called receptor. So plasma proteins are just carrier of hormones or carrier of drugs, or we can say they are drug binding. They are binding to the hormones and many substances. But plasma proteins do not produce any, once a substance binds with the plasma protein, usually plasma protein do not produce any specific response. So what I want to hammer in your mind, that to be a receptor, it should have minimum two domains, two special points at its number one, two special qualities. Number one, ligand binding domain and effector domain, and effect is produced. Is that clear? Right. Now there are many types of receptors. Right? We are, again I'm saying that we are concentrating only on the receptors in biochemistry and pharmacology, physiology. Right? Now, receptors, are many types of the receptors. I will just talk about their broad categories, and then I will go back in detail of G protein coupled receptors. Yes, you have a question.
Yes, sir. Actually, you said that the receptor binds and then produces a biological response. So will this response be limited to the cells only, or will it go through the whole system?
I'm going to tell you. Actually, he asked me a very important question. He says that when ligand bind with the receptor, and I'm saying that there is a biological response. Is this biological response only on cell? Answer is that if it is a cellular receptor, if it is on cell membrane, or receptors in the cytosol, or receptors in the nucleus, then of course there will be change in the cell. But if many cells undergo changes, of course that may produce some biological response which might be cellular or might be systemic, right? Multisystem, like adrenaline, epinephrine, norepinephrine. When epinephrine or norepinephrine is injected in the body, or dopamine, their receptors are present on multi organs and multi systems. So they will bring the changes in cells of multiple organs and systems, and multi-systemic response can come. So minimum receptor upon binding with the ligand can produce cellular changes, and maximum it can produce multi-systemic biological changes. It depends on what is the ligand and what is the distribution of the receptors on the tissues and how they are responding. Is that right? Yes. Let's come back that these receptors, where they are located. Let me make a cell here. Suppose this is a cell. Now, okay, let me make it a bigger cell. Let's suppose this is cell membrane, and here is the nucleus, right? Now, what could be the location of the receptors? There are some locations almost every student know, and there is one location usually student forget, right? Let me tell you the common locations. Number one is cell membrane. Some of the receptors are macromolecules, usually proteins with some oligosaccharides which are present in the membrane. We call them transmembrane receptors, or they are integral proteins, right? Then there are some receptors which are in the cytosol, right? And there are some receptors which are present in the nucleus, right? And there is one more site where their receptor can be. These are cellular receptor. The cellular receptor might be on the inserted on the membrane, or appearing on the membrane, or they may be in the cytosol, or even they might be in the nucleus. I will give you examples. But tell me one more site, any one of these front boys. Yes, any more site where receptors can be present. Where receptors can be present. That is, yes, come on. That is extracellular site. Some macromolecules are outside the cells, or you can say in extracellular space, or in between the cells, and some macromolecules are outside the cells, and sometimes some substances bind with them, bring the change into that molecule due to effector activity and change biological system. You must be thinking, what is this? You must be thinking, but I will not tell you now. First I will tell you these common three things, and then you have to ask me, remind me to tell extracellular receptors, right? Okay, let's start with general categories, main categories of membranous receptors, right? Membranous receptors are number one, yes, one-pass receptors. One-pass receptors are having ligand binding domain outside, effector domain inside, and one time they pass through the membrane, right? So this is a peptide. Let's suppose this is a peptide where it binds the ligand outside the cell, right? Ligand binding domain is extracellular and effector domain is intracellular, and one part is passing through the membrane, which is called transmembrane part. So this receptor has one ligand binding domain, one effector domain, and in between there is transmembrane component. So transmembrane component outside bind with ligand binding domain, and inside it is having effector domain. So when ligand bind with extracellular area, intracellular start some activity or some biological change or some signaling inside. In this way, these receptors, which we call them transmembrane receptors, this whole group is responsible for taking the extracellular signals, right? And under the signals, they bring intracellular change. They bring biological response inside the cell, right? They bring alteration in the cell. Now this is how many pass? One pass receptor. So we can say this is one pass receptor. Classical example of one pass receptors are tyrosine kinase receptors. Insulin receptor, yes, very good. Insulin receptor is one pass receptor. But let me tell you something interesting. When insulin binds with receptor, other receptor also come. So two receptors with one insulin, two one pass receptors bind with that, and that is dimerization. That, for example, this is one pass receptor. If insulin bind here, it will also bring one more one pass receptor, right? And two one pass receptors will make a dimer, and that dimer will bring intracellular changes on the target cells. But anyway, this is an example of what? One pass receptor. Then there are receptors which are usually, of course, they consist of peptide chains. Most of the receptors which are on the membrane, they are peptide chains. One peptide chain or more than one peptide chain. There are receptors which are not one pass receptor. There are more pass receptors, like 4 to 5 pass receptors. There can be 4 to 5 pass receptors. There may be 4 or 5 different peptide units which are going through the membrane, or there is one peptide unit but passes through 4 or 5 times. Can you tell me some example of such receptors? Yes, doctor. Any receptor where peptide unit is passing through the membrane 4 or 5 times, right? Are you going to tell me? Yes.
Sir, ion channel.
Ion channels, very good. For example, ligand binding ion channels. Let's suppose this is, yes. Let's suppose this is cholinergic receptors. Cholinergic nicotinic receptors. Acetylcholine will bind here. Acetylcholine will bind here, right? So this will be ligand binding domain, and when it binds here, let's suppose it changes the frequency of opening and closing of this receptors. So this tunnel through which this tunnel opens and closes, this canal which opens and closes, this canal is basically, yes, this canal, okay, let me tell you exactly, this is acting as what? Effector component. So upon ligand binding, right, upon ligand binding, some ion channels undergo conformational change. and that alters the flux of movement of the ions, right? So we call such receptors as, yes, ligand, yes, no, these are ion channels, but they are operated by ligand binding. So ligand gated ion channels or ligand operated ion channels. Such ion channels where function depend on the presence of the ligand. And effector unit is basically a canal, right? A central pore which is opening extracellularly as well as intracellularly, and when ligand binds, basically opening and closing of frequency of these canal changes, and that brings the biological changes, right? Then there are, so this is how many? 4 to 5 pass receptors. Then we come to our beloved receptors, the most loved one receptors. Even though they look like snake, serpentine receptors, they pass through the membrane 7 times. We call them 7 pass receptors. And let me draw here. Here is 7 pass receptor, let's suppose. This is one peptide. It is just one peptide chain, but it passes through the membrane 7 times. Now, because this peptide chain is passing through the membrane 7 times, 1, 2, 3, 4, 5, 6, and 7. Due to this reason, simply they are called 7 pass receptors. When they were discovered initially, that researchers found there are peptides in the membranes which pass through the membrane 7 times, and they thought when a ligand bind here, some changes are in the cell. So they said that this is 7 pass receptors, right? In those days, they only knew, for example, if adrenaline binds here, right? This change in, for example, cyclic AMP levels. Doctors, there was, so if catecholamines, adrenaline or noradrenaline bind with its 7 pass adrenergic receptor, there is intracellular change in cyclic AMP levels. In those days, many decades back, in between what was happening, we were not knowing. We were just knowing this thing, the adrenaline bind with the ligand binding domain, and somehow in the cell, what happens? Cyclic AMP levels change. Is that right? So then, doctors called adrenaline or such molecules first messenger, or molecules which were changing in the cell, which doctors were able to measure, they called that second messenger. That is why neurotransmitters or hormones or drugs, they are actually, when they go and bind with the ligand binding domain, we say first messenger has arrived. And ultimate biological change which is coming into cell, right, in the special molecules, we call them second messenger. But now, over the years, we have discovered there are so many things in between. For example, when ligand binds here, right, this is ligand binding domain. And what is this domain? Transmembrane domain. And what is this domain? Effector domain, right? Now we have discovered, again I will repeat it, what is it? What is this component? Ligand binding domain. And what is this component? Effector domain. This is effector domain, and this is ligand binding domain. And this peptide has one pocket outside where ligand can bind, specific ligand, and one tail inside, right? Usually this peptide has amino end outside and C terminal inside. Is that right? You know, peptide chain has 2 ends, amino end and carboxyl end. So what happens? These peptide chains, when they passes through the membrane 7 times, they are always keeping amino end outside and they are keeping the carboxyl end inside. So we say amino terminus is outside, extracellular, and what is this terminal? Carboxylic terminal inside. Is that right? Am I clear? Now, here effector domain is when ligand binds here. Now we have discovered that this effector domain does not change the cyclic AMP levels directly. In between there are many molecular signaling pathways going on, right? But when ligand bind here, there is a change in its internal part, right? For example, this will twist around. Rather than this form, it will change into this form. And its effector domain is exposed, right? Previously when it was like this, its effector domain was not exposed to the target proteins. But when ligand bind here, it undergoes a change in such a way that effector part become more exposed to the next generation of proteins which will interact, right? So we say effector upon binding with the ligand with the 7-pass receptors, yes, on the ligand binding domain which is extracellular, leads to conformational changes in all transmembrane components in such a way that intracellular component become exposed to the signaling pathways, right? For example, a special protein which will bind with this 7-pass receptor, there is a group of proteins, we call them G proteins. Here I want to remember, just mentioned G proteins are two types of G proteins. They are monomeric G proteins, monomeric G proteins, and there are, yes, trimeric G protein, trimeric G proteins. First of all, what is G protein? G proteins are those proteins which bind GTP or GDP, guanosine triphosphate or guanosine diphosphate. What are G proteins? G proteins are such proteins which can bind with GTP or GDP, right? Now, if G protein consists of only, you can say, yes, if it consists of only one peptide, then G protein is called monomeric. But if G protein has 3 peptides, right, all of them are making, now what is this? These are 3, 1, 2 and 3. 3 peptides together, we call it trimeric protein. You must be knowing that they are named as alpha component, beta component and gamma component, right? So, G proteins can be monomeric G proteins, G proteins can be trimeric G proteins, and the G proteins which interact with the 7-pass receptors, they are actually trimeric G proteins, not monomeric G proteins. Monomeric G proteins are having different signaling pathways. They do play a role in signaling in the cells. But when we talk about 7-pass receptors, we are talking about those receptors that upon binding with the ligand extracellularly undergo conformational change, and intracellularly, yes, effector domain changes in such a way, effector domain changes in such a way that trimeric G protein will bind there. What will bind there? Trimeric G protein. This is alpha component, right? And here is, let's suppose, beta and gamma component. So basically, they expose a site. Of course, when it was, if you remove the ligand, this will go into this configuration. And if it is in this configuration, and what do you think now G protein can bind here? No. So when ligand is removed, right, then effector domain become altered in such a way that it is not available to the trimeric intracellular G proteins. But 7-pass receptors, whenever they bind with the appropriate ligand, which is able to stimulate it, able to stimulate the receptors, that will eventually lead to change in the molecule in such a way that intracellular part, which is effector domain, is able to, or exposed in such a way that it is able to interact with G proteins. Trimeric G proteins. Don't say just G proteins. Say trimeric G protein. That is why I said monomeric G proteins have nothing to do with this receptor. Right? Is it clear? Now we know that whole signaling pathway we will discuss later. I'm just discussing about the broad categories of receptors. First category we are talking about those receptors which are expressed on the membrane and they have component passing through the membrane. We call them transmembrane receptors. We said there are, yes, one pass receptors. Classical example is for many growth factors or insulin, there are one pass receptors. Then there are 4 to 5 pass receptors which are ligand operated ion channels, right? Then there are 7 pass receptors, right? These are the most important receptors. Why? Because presently the drugs which are available in the market, right, about 30 to 40% drugs act on modifying the actions of 7 pass receptors. So it's very important to understand these receptors, their types and their signaling mechanisms and what biological responses such receptors produce. Of course, we will discuss in detail later. We are just classifying now. So we have here 7 pass receptor, which are also called serpentine receptors because peptide passes in and out through the membrane in a snaky way 7 times, right? So this is 7 pass receptors. Then there are another group of receptor and mostly students are not aware of that type of receptor. Another type of receptor present in the membrane. Yes, anyone from here? There are? Yes, from there. 12 pass receptors. Very good. There are also 12 pass. I don't know, I shouldn't make more. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 and 12. Something went wrong because, okay. So this is 12 pass receptors, right? These 12 pass receptors also consist of one single peptide chain, right? And this single peptide chain snakes through or passes through the membrane how many times? 12 times, right? Anyone can give me example of such receptors? One pass receptor, most simple example is insulin or growth factors, right? 4 to 5 pass receptors are ion channels, ligand operated ion channels. 7 pass receptors are very commonly for glucagon or adrenergic receptors or some dopaminergic receptors or muscarinic receptor for acetylcholine. Remember, for acetylcholine, cholinergic receptors which are ion channel, they are also nicotinic. Cholinergic receptors are divided into 2. Cholinergic receptors may be nicotinic cholinergic receptors or muscarinic cholinergic receptors. Nicotinic cholinergic receptors are ion channels and muscarinic cholinergic receptors are G protein coupled receptors or 7 pass receptors or serpentine receptors, right? Let's move on. And we were talking about that there is 12 pass receptor. 12 pass receptors. Yes. Do you know any example of 12 pass receptors? I'm not happy with these all researchers. They keep on bringing the new things. It was good to be doing the graduation in my time. There were not so much knowledge. In your time, too much knowledge. But people who love knowledge, it's very lusty development. More and more knowledge. Okay, so 12 pass receptors. These receptors are usually present on such neurons which release monoamines. Suppose this is a neuron which releases dopamine or epinephrine or norepinephrine. For example, it is releasing norepinephrine. Now, on the nerve ending, there is, what is there? 12 pass receptor. Right? 12 pass receptor. What is the function of this 12 pass receptor? That, of course, this norepinephrine will act on its receptor on the postsynaptic membrane, and then some of this norepinephrine is reuptaken back. This reuptake protein, right? Norepinephrine or dopamine or serotonin, when they are released, right, they go and act on the postsynaptic membrane, produce the biological changes, but because these are very precious ligands, most of it is taken back. And because they are taken back, so they are not lost, we say that they are reuptaken in the presynaptic membrane and again restored, right? So this reuptake protein is like serotonin reuptake protein or norepinephrine reuptake protein or dopamine reuptake proteins. These are basically what? That, for example, serotonin bind here, it undergoes the change, functional change, and throws the dopamine inside. You get it? So what is this? This is the receptor. And it passes through the membrane how many times? 12 times. So 12 pass receptor, one of the classical example is those receptors present on the presynaptic membrane, right? On the yes, monoaminergic neurons, nerve endings, which release monoamine neurotransmitter. Monoamine neurotransmitter means neurotransmitters which are derived from one amino acid, like epinephrine, norepinephrine, serotonin, dopamine, right? They are monoaminergic neurons as a group. Noradrenergic neurons, adrenergic neurons, dopaminergic neurons, serotonergic neurons. These are all monoaminergic neurons. And this is monoamine reuptake system. And this reuptake system consists of special transmembrane proteins which are 12 pass receptors. Is that clear? So how many transmembrane proteins are types of receptors are there? 12 pass receptors. So we have 1 pass receptor, right? We have 4 to 5 pass receptors, right? And we have 7 pass receptors, and we have 12 pass receptors. So these receptors are present on the membrane. Just rapidly tell me. Yes, what is it? 1 pass receptors. What is, might be this? 4 to 5 pass receptors. What it could be? 7 pass receptors. And what could this be? 12 pass receptor. I have not counted it anyway. Right? So these are classically broad categories of receptors which are present on the membranes on the target cells. Right? Then receptors might be present in the cytosol. Okay, they might be present intracellularly also. Now, why some receptors are present intracellularly and why some receptors are present on the membrane? Answer is very simple. Those ligands which cannot cross the membrane, they should have receptor on the membrane. Those ligands which cannot cross the membranes, they must have receptors on the membrane. So usually these receptors which are on the membrane, their ligands are either highly polar or molecularly large. So if they are large molecule or highly polar or charged molecule, they cannot pass through the membrane. As they cannot pass through the membrane, like epinephrine, norepinephrine, dopamine, serotonin, histamine, these are charged molecule. They cannot cross through the membranes. So their receptors should be on the cell membrane. In the same way, if we talk about insulin or glucagon or follicle stimulating hormone or luteinizing hormone, these are peptides. Peptide also cannot cross, they have significantly larger molecule, simply they cannot go through the membrane, so their receptors should be present on the cell membrane. But there are some substances which can cross the cell membrane and go inside. It is very natural if someone cannot come inside your house and he has to give some message, he will knock on the door. But some member of your family will simply walk in. Right? Similarly, some ligands can simply walk in, diffuse inside the cells. For example, these substances are either very small or they are lipid soluble. So they can pass through the lipid bilayer. Right? Classically here I can say like estrogen receptor. Estrogen can go inside the cell. In the same way, T3, thyroid hormone, Now estrogen can easily go inside the cell, or tyrosine is significantly small molecule, can T3 is significantly small molecule, can go inside, and estrogen, of course, it is derived from the modifications over the cholesterol molecule, which is lipid soluble, can go inside the cells. So such molecules, or even vitamin D3, right? So such molecules, their receptors are where? Intracellular. Now these intracellular receptors, right, for example, for estrogen and T3, if we focus just for these two examples, they can be present in cytosol, or they can be present inside the nucleus, and the receptors are present on the DNA. The receptors are DNA, right? For example, DNA act as receptors for alkylating agent, anti-cancer drugs. Now, estrogen has receptors. Okay, I will just try to confuse you. Let's see what happens. You get confused or not. Let me try. Estrogen receptors is in cytosol or in the nucleus. Actually, estrogen receptor is cytosolic receptor or intranuclear receptor. And other question is, T3 receptors are primarily located in the cytosol or inside the nucleus. Do you have an answer? Yes. My question is very simple, that estrogen receptors and T3 receptors, both of them are intracellular. One of them have receptor in the cytosol, other has receptor in the nucleus. How you can guess it? It's very simple. This is coming from thyroid. When you talk about T3, what do you think of? Which gland? Thyroid. And when you talk about estrogen, the most visible sign of estrogen is what? Breast. Estrogen, progesterone act on the breast. Is that right? Yes. Clear? Yes. When you would think of estrogen, estrogen has many functions, but at least you should remember development of breast, right? And T3, whenever you think, your mind should go to which hormone? A gland? Thyroid. Now you tell me, breast, well, fully developed breast is bigger or thyroid gland is bigger? Normal thyroid gland is bigger or normal breast is bigger? Breast is bigger. So breast should be, now it is a little naughty thing, breast should be in the cytosol or in the nucleus? Cytosol. And if it is in the cytosol, of course it is not there. Just remember, so estrogen receptors are in the cytosol. Estrogen receptors are in cytosol, right? Just it is to remember. And T3, T4 receptors, especially T3, which is active form, its receptor should be nucleus, right? T3 receptors, right? So what we have discussed, that there are receptors which are cellular receptors and extracellular receptors. Cellular receptors may be transmembrane receptors or membranous receptors, and there can be cytosolic receptors or there can be nuclear receptors. Classically, they are broadly speaking, there are four categories of membrane receptors. They are 1-pass receptors, 4 to 5-pass receptors, 7-pass receptors, 12-pass receptors. And when we talk about intracellular receptors, there are so many examples, but we just talk about two, estrogen receptors and T3 receptors. Estrogen receptors are present in cytosol and T3 receptors are present in nucleus, right? And easy way to remember is that usually fully developed breast is larger than fully developed thyroid gland. So cytosol is a bigger space which can accommodate the breast. It is just your imagination. And nucleus is smaller space which can accommodate thyroid gland. But actually we are talking about referring to not thyroid gland, we are referring to T3 receptors. Here we are really not talking about putting the breast in cytosol, we are talking about estrogen receptors in cytosol. When estrogen comes, it will meet its receptor in cytosol on the target cell, then receptor estrogen complex will move to the nucleus and alter the expression of genes. Is that right? Now we are left with the last category of receptors, which are extracellular receptors. It means there are some macromolecules or complex of macromolecules which are outside the cells in extracellular space, and they also do respond to ligands and bring biological changes in the system, right? So you can tell me any extracellular receptors? Yes, doctor.
Sir, heparin receptor.
Okay, actually coagulation factor we can say. Coagulation factors, many drugs interact with coagulation factors and alter the function of the coagulation factor. So coagulation factors are extracellularly, they are present in plasma, is that right? In the blood. Or for example heparin act on, yes, antithrombin 3. So that is extracellular proteins, right? The function of antithrombin 3 is that it inactivates some of the activated coagulation factors, especially activated 10 factor, right, and some other. So actually what happens that some of the coagulation factors, of course, coagulation factors are outside the cells, and some drugs binds with coagulation factors in such a way that coagulation factors function is altered. Either their function is prolonged or their function is reduced, or what happens, let me tell you, suppose this is antithrombin 3. What is this? Antithrombin 3 molecule, right? This is extracellular, and here what binds here? Heparin. When heparin bind on this point of antithrombin 3, this is ligand binding domain. But when heparin bind over here and disturb the tail, it must become more active to destroy the coagulation factors. This is active coagulation factor, and here are pieces of it. So what happens? Heparin binding domain is present on antithrombin 3, and this is ligand binding domain, heparin binding domain, and this is effector domain. So once the ligand or heparin binds with the, what is this? Antithrombin 3. Antithrombin 3 molecules capability to inactivate the activated coagulation factor is enhanced. So this must be effector domain. You are getting it?
Yes, yes,
So what we can say, some of the coagulation factors which can bind with the drugs or bind with the certain you can say endogenous substances and undergo altered function, we can consider them as receptors. Like antithrombin 3 is considered receptor for heparin. In the same way, some drugs act on the activated complement proteins like C5a. Some drugs act on C complement 5a, right, and alter the function of C5a. So that will be what? Extracellular receptor. In the same way, there are many drugs now coming which act on the antibodies. There are many drugs which interact with the antibodies and alter the function of the antibodies. Let's suppose this is an antibody. Suppose drug binds here and alter the function of antibody, right? So in this case, this will be ligand binding domain, this will be effector domain. You are getting it? So what we can say, that actually some of the receptors or macromolecules are present outside the cells. We call them extracellular receptors. These extracellular receptors, right? These extracellular receptors, right? Classically, they may be coagulation factors, or they may be, yes, antibodies, or they might be cytokines, very good, cytokines, or they might be activated C5a, right? So drugs which are acting with these molecules, then bringing the changes in them, in their functional changes, right? We say that drugs are acting as ligand, and these proteins, extracellular protein, when bind with the ligand, they produce a biological response, altered response. So these proteins are behaving as receptors. Let's sum it up. What is a receptor? Receptors are basically two types. They are sensory receptors and biochemical or pharmacological receptors. Sensory receptors are related with central nervous system and picking up different sensations. We will not talk about that today. Today we will focus on which kind of receptors? Pharmacological or biochemical, or receptors in biochemistry, or receptors in the physiology, or receptors in pharmacology, right? Especially this group of receptor, we call them that these are all macromolecules. Usually they are protein, but they may not be protein. For example, some act on DNA. So DNA is a macromolecule. What is a receptor? Receptor is a macromolecule or a complex of macromolecule which should have at least two functional domain, ligand binding domain and effector domain. Is that right?
Yes, sir.
Number one. Number two, receptor types and sites. Receptors are present cellularly and extracellularly. Cellular receptor first, because they are more abundant. Cellular receptors are membranous receptors, which are usually transmembrane proteins, and then there are cytosolic receptors and there are intranuclear receptors. Classically speaking, membranous receptors have four broad categories. Yes, 1-pass receptors, 4 to 5-pass receptors, 7-pass receptors, 12-pass receptor. Very good. These are membranous receptors, right? 1-pass receptor, classical example is like for insulin or like for different growth factors. 5-pass, 4 to 5-pass receptors are usually ion channels, which alter their function of binding of specific ligand. And 7-pass receptors are intracellularly coupling with G proteins. So 7-pass receptors are also called G protein coupled receptors, right? They couple with the trimeric G protein, not with monomeric G proteins. They couple with trimeric G proteins, not with the monomeric G proteins. And after 7-pass receptors, we come to what are these? 12-pass receptors. 12-pass receptors are found especially in those new nerve endings, presynaptic nerve endings, those nerves which are monoaminergic, right? Minergic nerve endings which release epinephrine, norepinephrine, or dopamine or serotonin. What is the function of this 12-pass receptor? They act as reuptake mechanism for the released neurotransmitter, right? Some of the neurotransmitter will diffuse away and that will be catabolized somewhere in the body, but much of it will be reuptaken, right? And why I call it receptor? Because some drugs can work over here. Number one, for example, serotonin can bind here, 5-hydroxytryptamine, that can bind here, and that is taken from extracellularly, transported by the receptor intracellularly, and might be it is stored again. Number two, some drugs can bind with these receptors, serotonin reuptake inhibitors like fluoxetine. So fluoxetine is working on what kind of receptors? 12-pass receptors, right? So let's take a break and then we'll continue. Any question you have?
Sir, we have discussed that T3 receptors are intranuclear. What about T4 hormone?
Actually, most of the T4 hormone, before it enters into cell, is converted into T3, and T3 is the active form. So we are more concerned about T3 receptor. Is that right? Thank you very much. Class dismissed. After this, in the next lecture, we will be talking about in detail G protein coupled receptors, and then of course G protein coupled receptors, ligand binding domain in the next lecture. Especially I will define and differentiate agonist, antagonist, which are easy concept, and real concept which I will explain is partial agonist antagonist molecules. Partial agonist antagonist molecule, right, or drugs. Then I will talk about transmembrane domain, effector domain, actions and functions of, yes, of 7-pass receptors, that how they activate the G proteins, what is the exact mechanism, how G proteins bind with the effector domain of 7-pass receptors, and then I will discuss very important thing, how these receptors can be inactivated, 7-pass receptors, or they can be desensitized or resensitized or downregulated or upregulated. We will talk about these concepts in coming lectures. Class dismissed. Thank you very much.